All right. So yeah, it's great to be here with everybody. For some reason I can't see my camera. Sorry, I'm just getting myself sorted in my setup at home. As Walter said, we're now going to take over for the next 8 hours of the meeting. So we've got my co-moderator, who is Wei Xu, and myself. I've just joined La Trobe University, so I'm in Melbourne. As Walter said, we're in lockdown at the moment, unfortunately. So I'm sorry if you hear a bit of background noise where I am. Wei is in Perth at Murdoch University. Wei, do you want to say hello and maybe introduce yourself?
Hi, everyone. Thank you. My name is Wei. Currently, I'm a senior lecturer in Murdoch University in Perth, in Western Australia. And previously, I did my PhD in UC Davis in Dr. Walter Leal's lab and start the insect olfaction work. And then I moved to Australia to do a postdoc in CSIRO. And 6 years ago, I moved to Murdoch University to start my lab. Thank you, everyone.
Thanks, Wei. And for people who don't know me, so my involvement in the field of insect taste and olfaction goes back 20 years, where I did a postdoc with John Carlson at Yale, and I was involved with Peter Klein in the work identifying insect odorant receptors and taste receptors. The first in a big race with Leslie Vossel, who's here, and others. It was a fantastic time. I've worked on a lot of other things since, but maintained a strong interest in the field, and it was really lovely to be invited by Walter to host this section of the meeting. So we're going to, as Walter said, we're now going to take you through the next 8 hours. We've had Kay Dong just present to us as a transition into our section. We've got a couple of other keynote speakers throughout the day, Gurong Wang and Melissa Jordan. And then we'll have Richard Benton, who will transition us through into the British Standard Time section of the program that Wynand will host. We also got in our section 15 contributed presentations, and we're going to start with some who are actually still based in the United States. And then we're going to move west, and we have speakers from New Zealand, Australia, China, and Japan, and India. And then the last few speakers of our section will actually be in Europe. We have some speakers. We have David Heckel in Germany. We have Alice French in London. And then we'll have Richard Benton in Switzerland as our bridge into the British Standard Time section. Again, just a reminder, as Walter said, all the attendees, we can't see anything you put in the chat, and we can't see you raise your hand, so you need to use the Q&A feature for your questions. You can make them anonymous if you would like, and if it's helpful if you are a student or an ECR, you can tell us and we will happily prioritise your questions for you. Okay, so I'm now going to hand over to our first contributed presentation for our section, which is from Teruyuki Matsunaga. Teruyuki, if you could share your slides. He's going to talk about evolution of olfactory receptors tuned to mustard oils in herbivorous Drosophila. Thanks, Teruyuki.
Sorry, I can't share the screen. That's weird.
Sorry, I have to stop. You go, try now.
Yeah, okay, good. Okay. Okay. Yeah, thanks for the introduction, and I'm really honored to be able to be part of it. And yeah, especially I'm Japanese, I'm from Japan, so I really enjoy the Kedon's talk, the Chris Hanseman. So yeah, yeah, I really like that presentation. Thanks so much. So let me go to the introduction. If we go out field, you will find so many insect species, so many plant species. And these diversities are propensified by the arms race between the insects and the plant species. So especially the herbivory. The herbivory means the insect feeding on the plant species. That is one of the most important part if you think about the diversification. So how the herbivory started, and when it started, it's dates back really old. Like for example, moths, butterflies, they date back over 100 million years ago. Also, same for the beetles. So it's like the age of the dinosaurs. But if you look at this lineage Scaptomizer, they are relatively recent, 10 million years ago. It's more like the chimpanzees or mammal species. So if you want to dissect how the herbivory started, it's a good system to use the Scaptomizer. And we specifically use Scaptomizer flavor who likes to eat the mustard plants. This is a female Scaptomizer flavor who had a serrated ovipositor, then cuts the leaves and then drink the juice from the leaves. That must be so bitter for human, but they like it anyway. So if you look at the phylogeny, this is the simplified phylogeny that includes three species, including the Scaptomizer flavor, Scaptomizer pallida, and Drosophila melanogaster. As I said, the flavor, they like the fresh leaf of mustard plants. On the other hand, the Scaptomizer pallida, the close relative of flavor, they like the rotting plant materials or the microbes. This is this niche is relatively similar in the Drosophila melanogaster. So the Scaptomizer pallida, Drosophila melanogaster, they kind of share the similar niches, but the Scaptomizer flavor, they have the drastic difference of the niches, which feed on the fresh leaves and the mustard leaves, as I said. The mustard plant, the Brassicaceae, family Brassicaceae, it has these species, wasabi, black mustard, arugula, radish. If you think about that, if you eat them, maybe you feel pungent sensation. And that is mediated by the chemical compounds called the isocyanates, ITCs. And this ITC is very rich in diversity. If you change this chemical compound, the residue, you can change so many, you will find so many ITC species. There is over 100 species already found in ecology. And as I said, it's very, very pungent. So if somebody make the paste of the wasabi and try to rub under your nose, you will freak out, you will be really mad. But the Scaptomizer flavor, weirdly, they really like it. And we hypothesize maybe they are attracted to the ITCs. So these are the questions we addressed in this study. The first question we asked is the flavor attracted to ITCs? And if so, which odorant receptors mediate detection of the ITCs? And if we express that flavor-derived ORs in microfeeding Drosophila melanogaster, are they attracted to the ITC eventually? Is the question we addressed. So first of all, the flavor attracted to ITC. We use the Y-maze dual choice assay. Here the control versus order the flies can choose. And we try these three species, and the horizontal here shows the percentage of the flies choice to the order versus to the control. For example, the apple cider vinegar, which is known the strong attractant for the melanogaster, attracted the melanogaster. So this is consistent with the previous results. If you look at the arugula, the mustard plants, that attracted the Scaptomizer flavor. And as I said, the ITC is rich in diversity. So we just use two types of the commercially available ITCs here, and the BITC, SBITC both attracted Scaptomizer flavor. So now we know the flavor is attractive to the ITCs. And the next question is which ORs? We screened the ORs out of around 300 ORs in total using these 5 genomes that time it's available when we screened. That is the flavor Grimshawi mohabensis viris melanogaster. And the criteria we use are here too. If the ORs duplicated and also the DNDS is elevated. DNDS is defined by this. In short, if the DNDS is high, that means the signature, there is a signature of the positive selection. And here is the gene tree. The green branches show the Scaptomizer flavor. And if you look at this lineage, the OL67B, that is interesting because it has the triplication in a Scaptomizer flavor lineage specifically, but not the other species, and also the DNDS was elevated. If you extend when we extended the species, the 4 Scaptomizer species and also the 12 Drosophila species, we find similar consistent results. The DNDS indicated red or blue. The Scaptomizer flavor or 67B3 copies were always, the DNDS were elevated. So OR67B is a good candidate, and we want to know what the function is. So we use the empty neuron system available. That is published and a lot of people used. That is the Helen et al. 2005 and Kutovic 2007. And we basically express the OR67B from the several species and also the paralogs of the flavor in the deletion mutants of the ORs. And we path the orders and try to see which odorants activates these orthologs or paralogs of the OR67B. And here is the representative results. We of the Scaptomizer pallida OR67B and the Scaptomizer flava OR67B1. And we tried the acetophenone compounds and the BITC. Acetophenone is already known to activate the Melanogaster OR67B. So we chose this, and here is the raw data. For example, acetophenone very strongly activates the pallida OR67B, but not in the flava OR67B1. On the other hand, BITC activates very strongly for the flava OR67B, but not the pallida OR67B. And here are all of the plots by the chemicals by the known OR67B activator for the melanogasters, and also the ITCs. And as you see, the melanogaster activators consistently activates the melanogaster OR67B, consistent with the previous results. And pallida OR67B is a little bit less strong compared to the melanogaster OR67B, but still activated, especially compared to these other flava OR67B 1, 2, 3. On the other hand, ITC only activates these flava 67B 1, 2, 3 paralogs. So this indicates the ancestral states of the OR67B are, like here, the ancestral states are most likely something like these tuning curves activated by, for example, acetophenone. But the ancestral state of the flavor OR67B around here, maybe they lost the sensitivity to these ancestral states. And then now instead they gain the ITC sensitivity. So the next question is, like, why they have the 3 replicates or 67B 1, 2, 3? Maybe there is some functional difference. We tried dilution series to address this idea question using several ITCs, and I included just 3 here. OMB ITC mainly derived from arugula, AITC from wasabi, or PITC from watercress. And this is a dilution, different concentration. And as you see, the B1 was strongly activated by the 4MB ITC, same as the AITC, PITC, respectively activates the B2 strongly, most strongly, and the PITC most strongly activates B3. So these paralogs are similarly activated by the ITC. By ITCs, but there is some functional difference between them. So far, this is a summary of the results so far. So the ancestral state of the OR67B, it is activated, most likely it is activated by these ketone esters alcohol, including the acetophenone, but this chain, they split here, and then at the B, the ligand changed, the peak shifted towards the ITCs, and then finally this OR67B triplicated and having the different sensitivity to the ITCs. So this is the evolution of the OR67B. The next, we are interested how is there any evolution of the behavior In order to ask this, we express the flower-derived OL67B into the melanogaster. So usually, wild type, the melanogaster has the OL67B tuned to the, for example, acetophenone, and they are attracted to the rotting foods. Now we express the flower-derived OL67B tuned to the ITCs, and is the melanogaster attracted to the ITCs. And so the result is here. The genetic controls, they didn't really show the preference. They were kind of neutral. And when we express the melanogaster OL67B or flavor OL67B3, only the flies who carry the OL67B3 in the melanogaster neuron that usually express the OL67B derived from the melanogaster is attracted to the ITC. So, yeah, by the way, this is accomplished by the Y maze. So yeah, we found now these flies are attracted to the ITC. The mechanism underlying this is probably most likely one hypothesis is that the melanogaster OL67B expressing olfactory sensory neuron mediates attraction. In order to ask this, we silenced the melanogaster OL67B neuron. So yeah, usually it looks like this. The melanogaster is attracted to the, it's already known, low concentration of the acetophenones attracts the melanogaster. And try to see if the OL67B neuron silenced, is the attraction to the acetophenone is lost. So here's the data. First of all, we saw the trend of the attraction towards the acetophenone in a genetic control flies. But when you silence the OL67B neuron by KIR, the flies even show the aversion to the acetophenone. That indicates the OL67B olfactory sensory neurons in the melanogaster mediates attraction. So yeah, we hypothesize, this is the conclusion and the takeaway. So the melanogaster OL67B is tuned to the acetophenone and foods attracted to the rotting foods. And now the flower OL67B is triplicated and also changed the tuning patterns. Tuned to the ITC, different types of the ITCs, and now the flies are attracted to the mustard plants. So the takeaway is that the chemosensory specialization in herbivorous insects can likely result from the relatively simple genetic modification at the periphery. So yeah, I want to acknowledge the people who contributed to this research, especially postdoc advisor Noah Whiteman. He's very, very warm person, and everybody here, the Carolina especially led the behavior assay with Hero, and yeah, everybody contributed. So thanks so much. And every lab members in Noah Whiteman's lab and the funding sources. So that's it. Thank you so much for your attention. And I'm happy to address any questions or comments. Thank you so much.
Thank you, Teru, and thank you also for agreeing, despite being in the US, to speak in our time zone. I think that's really helpful for us as well. Do we have any questions? And I think who is sharing their screen at the moment? Is that still your screen, Teru?
Okay.
Thank you. Thanks for that. So do we have any questions either from the attendees or from the panel members? I might ask one while we're waiting. Did you mention early on in your talk that there were about 300 OR genes in this species? Did I catch that correctly?
Sorry, say that again. 300.
Did you mention early on that there are about 300 OR genes in your species?
So each species has approximately 6 ORs, and we use the 5 genomes of the Scaptomyza flava, including the Scaptomyza flava and the 4 other Drosophila species. So in total, 300. Sorry for the.
Yeah, no, that's fine. I just always find it really fascinating, some of the expansions that have happened in some of the species. So I was just trying to get a sense of whether they're overall there are an equivalent number of ORs to many of the Drosophila species.
Yeah.
Wei, would you like to ask your question?
Yes, so yeah, nice talk. My question is, where are these OR60B expressed? Only in the antennae? Did you have a chance to check like a maxillary pulp or ovipositor? And also, did you have a chance to check other fruit fly, not just Drosophila or like taffrated fruit fly? Yeah, other fruit fly, do they also have the similar receptor genes?
Thank you so much for the question. So first of all, where it's expressed, we did the transcriptome analysis and we know it's expressed in the antenna. We've never done the maxillary. We know that maxillary pulp, it's not expressed very strongly. It's how can I say? It could be detectable, but I doubt it's functional in the maxillary pulp, and it's originally in the I don't know if it's originally or not, but in the Melanogaster paper, it's already known it's expressed in the antenna. So, but not in the maxillary pulp. So we think not in the maxillary pulp expressed in the flavor either. We've never tried the ovipositor. I think that's very interesting idea. We probably should try if it's expressed. And also we randomly poked the antennal sensilla, and we found basiconic sensilla. So yeah, basiconic sensilla and a trichoid sensilla. There are some neurons that is activated by a set of the ITCs, and we think that is the OR67B expressed.
Great. Thank you, Teru. So I can't see any other questions coming in from the panel or attendees. Just check. All right, so we are running nicely on time now, actually, so that's fantastic. So we might move on to the next speaker. And I would just say it's lovely to see all the names coming and going, and now that we're seeing lots of attendees that I recognise from Australasia joining the meeting. But I appreciate all the people in the US who are sticking around as well. It's really kind of nice. So our next speaker, let me just share my screen. Is Brian Smith. So Brian is in the US at Arizona State University, and he's going to talk to us about early olfactory processing in the honeybee. So Brian, please take it away.
Well, thank you, Coral. Let me get set up here. And it's a real pleasure to be a part of this symposium. I want to thank you and Walter and Wynand for all the effort that you put into organizing. It was such a great idea to link us all around the world in 24 hours and have us all talk about some of the newest stuff that we're doing. And I wanted to back up a little bit from the title I submitted and try to give you a little bit of the big picture to show you where this new research we're about to try to publish fits into the bigger picture. They work with honeybees and bumblebees, and of course they're foraging on flowers that have these complex kinds of odors, and many times they have to make very subtle distinctions between a flower that has nectar and a flower that has a very similar but subtly different mixture that doesn't. And they come with an olfactory system that's enormously capable of making those kinds of distinctions. They have about 160 plus receptors in their genome, that many glomeruli in their antennal lobe. So when you think of combinatorial coding, the coding space is enormous in terms of what they can potentially represent. But the interesting thing that was always puzzling to me is odors that they need to, that this space can encode, are going to be irrelevant to any given bee at any point in its life when it has to make a decision about some very specific odors. So it comes into the world equipped with the ability to do it, but they're not using this space most of the time. And my lab over the years has been studying a variety of associative and non-associative plasticity in the antennal lobe. And the bottom line is what happens is this plasticity at this early level of processing sort of warps the space to expand it, to help the bee solve the problem at hand right now. Does this with a brain with about 1 million neurons. And I won't go specifically into insect anatomy, most of you know that, but I want to show this neuron, a VUM neuron, in particular, that fascinated me almost 40 years ago when I was a host in Randolph Menzel's lab. They just discovered this neuron. It releases octopamine all over the brain. It looks like a modulatory neuron, and it does that when the bee's feeding on sucrose, which is nectar. My late colleague Martin Hammerhead was at the time, we were colleagues then, he was performing some heroic electrophysiological recordings to show that this is probably the representation of reward. And I thought, well, if it's doing this up in the mushroom bodies here, typically the seats of learning and memory, what's it doing down here in the antennal lobe? And I want to tell you that story now. So the antennal lobe, the quick story here is that it encodes odors. A lot of the imaging that we've done, you see about the 20 or so glomeruli on the dorsal surface, and you start stimulating with odors, and you can see that different codes are set up to represent different odors. That's just the molecular level representation in the antennal lobe. Well, why do I think there's more to it? If you look at any individual glomeruli, the pink here are terminals of sensory axons, and the blue here are dendrites of projection neurons, and they're interacting here in the cortex of this glomerulus, and you can divide it up into the core and the cortex. Obviously, it's more than just a pass-through information system. You have GABAergic interneurons that are represented in the core and the cortex of any glomerulus. But when you start looking at things like octopamine, you'll see the terminals of the VUM neuron represented here in the cortex, where there's a lot of synaptic connectivity between inhibition and excitation. You see also serotonin distributed throughout the glomerulus, and then you see dopamine that's distributed, is not limited to glomeruli, but is distributed throughout rather broadly through the antennal lobe. So that's why we started to refer to a glomerulus and glomeruli as computational units, doing something very complex in terms of representing odors. The nice thing, as you know about bees, is they learn, they're brilliant. We bring them into the laboratory and we put them in these small restraining harnesses. You associate a little odor with sucrose. The bee sticks its proboscis out to the odor, so we can say, can it detect the odor and discriminate? I'm going to tell you a little bit toward the end about these antennal movements, which we've been tracking too. It's been known for some time that they can be very expressive. Well, you train a bee to an odor, and then you test it with a series of odors, either the odor you trained it to or something very different, as well as a variety of structure mixtures in between, and you establish these generalization gradients, and you can ask, can the bee discriminate odors? Another kind of protocol that I've worked with extensively is not just learning to pay attention to something, is learning to ignore things. You train a bee that this odor means nothing by presenting 40 or 50 trials followed by nothing, and then you try to train this odor with sucrose reinforcement, and they learn that odor slowly relative to a control odor that it didn't experience. So they're learning to ignore this orange represented odor right here. It's called latent inhibition. And it's a very important kind of learning. It's more complex than it looks. visual and olfactory components. We've worked quite a bit with this kind of learning. Fernando Locatelli, who may be here today, was in my lab many years ago, and he started doing imaging now, calcium imaging of these glomeruli on the dorsal surface. And of course, when the odor comes on, you can see over the course of time, this orange box here is when the odor comes on, and you can see the antennal lobe begin to respond and slowly shut down when the odor turns off. Well, if you take these snapshots here, 200 millisecond snapshots, and we're going to reduce 20 dimensions down to 2, principal components analysis, then you can represent these odors as a trajectory through this two-dimensional space where the little X's here that you might barely be able to see represent the snapshots. So the red is one odor here, the blue is a different odor, and then these are odors that are in between. So this represents a perceptual dimension as is represented in the antennal lobe. Well, Fernando, many years ago, would train bees to discriminate, say, one odor, one mixture from another one, and then we could do the calcium imaging and calculate the distance between these snapshots all along the trajectory, this transient that represents an odor. So you have naive bees, you have bees conditioned, this one's plus, this one's minus, and what happens with the distance? Naive bees, the distance when the odor is on, that Euclidean distance begins to increase. However, if the bees have been trained, one is good, one is bad, you get a much greater separation. So this is what I meant by the space is warped. It's stretched by the associative plasticity that Fernando has studied and published in a couple of papers. Now, likewise, if we generate latent inhibition, we teach a bee to, for example, ignore the blue odor right here. And so the blue odor is going to be the familiar odor. The red odor is the novel odor. And the imaging, these don't change. I'll tell a little bit more about the story in a few minutes using electrophysiology. But what Fernando was able to establish was that the 50-50 mixture of these two odors shifts to become much more like the naive odor or like the novel odor after you've presented this odor 40 times. So what antennal lobe is doing is it's biasing the representation towards something new. You want to ignore what doesn't mean anything and really make sure that you pay attention to the new thing. And of course, if we generate latent inhibition to the red odor, then the mixture shifts in the opposite direction. So this is now, I've shown you the instantiation of both associative and non-associative plasticity in the antennal lobe and space warp. Well, lately we've been working with antennal movements, and we noticed that bees, when you generate latent inhibition, you present an odor 40 times, they look very bored, by the way. And when you suddenly present them with a new odor, they get excited, almost as though it's a kind of novelty detection. And Chris Jernigan, then a PhD student in my lab, started looking at this. And if you look at the antennal positions here, so the antennae can be forward like this, and that will correspond to this position. The intermediate, the black antenna below, is this position, and the green is the antennae are being held back, almost in a position that the bees hold them when they're flying. You can see when the odor comes on, this is going to be the familiar odor. Both of these are naive, they're naive, so this is before we've conditioned them. The odor comes on, the antennae move back, and then the odor goes off and they move much more forward in this space. And they do that to both of these odors. Well, we familiarize the bee to this odor 40 times, no reinforcement. And then what happens is that backward movement of the antenna is very much diminished. They're not paying attention to it anymore. But look at what happens to this novel odor now. Once we've presented this one 40 times, the antennae go back and they stay back, almost as though the bee's searching for an odor because they're very excited. And this is what we've been calling novelty detection that's revealed in these very expressive antennal movements. Well, to look at this, we now been using multi-channel electrophysiology. Hong Lei in my lab has been doing that. And you impale the antennal lobe with these electrodes, and you can measure many different units, discriminate them by waveforms and firing patterns. And working with a colleague at UC San Diego, Seth Haney, we've been able to statistically separate these based on a couple of different characteristics into projection neurons and local neurons. And based on that, we've been looking at, well, how is this novelty detection potentially represented in the antennal lobe? So you look at how projection neurons respond. I'm not going to show you a lot of data with local neurons, but the story is largely the same. The response to control air puffs doesn't change pre-familiarization and post, that is before the 40 presentations and after. But the response to the familiar odor goes down. The response to the novel odor goes up. So this is the representation of novelty at the level of the antennal lobes. Happens both, as I said, for local neurons. And this interaction term in the ANOVA is significant. The t-test here isn't quite significant. Let's refine this analysis, because this is throwing every unit we're recording into the mix. That includes a lot of units that don't show much responsiveness to the odors we're using. And let's just separate the odors now by whether they're biased to octanone, which is one odor, or biased to hexanone, which is another odor. And we can select this based on a few criteria, not just firing rate. Well, when you look at when you pick projection neurons that are biased initially in their responsiveness to the odor that we're going to familiarize, the response to the familiar odor in these specialized projection neurons will go down significantly. The response to the novel odor goes up, and this, I think, are hexanol biased units, and octanone biased units do the same thing. So now Hong had the idea of counting just the number of cells that are above this baseline. This is, by the way, the difference of response before and after familiarization. So if the responses are the same, this is 0. So you count the units above and the units below and represent them this way. And you see that the familiar odor in the biased units is reduced both when it's hexanol and when it's octanone, and the novel odor increases. Units that are not biased toward the familiar odor don't show these changes. So it's really the units that are biased to begin with. And we think now that this is a different kind of associative, non-associative learning linked to habituation or latent inhibition. That is, you ignore things, and when something new comes along, you really pay attention to it. So I think with all of these kinds of learning we've studied, we've shown that these glomerular codes of odor are modified by experience. They're not changed dramatically. They're tweaked a little bit to warp that space, as I say. Both associative and mechanisms now tune these responses. So the antennal lobe can represent food or nothing or surprise expectations. And now we're adding familiarity and novelty to that learning. Ongoing, where and how is this novelty detection implemented? We don't yet. Is it in the neural network of the antennal lobe? Is it in the mushroom body? And there's some evidence in fruit flies that it could be implemented in the mushroom body network, could be implemented there and fed back into the antennal lobe. We don't know. The persistence of novelty effects through time looks like it diminishes quickly, but we haven't studied that yet. And then lastly, we're very interested in studying the role of novelty in driving foraging decisions in realistic, potentially virtual reality environments that we hope to start flying bees in the next few years. So we have a lot to do. certainly more than enough to take me to retirement in a few years. Had a number of wonderful people.
So if we could wrap up.
Yes, I'm wrapping up. Had a number of wonderful people doing a lot of this work. I mentioned a few of them. And I'd like to acknowledge lots of extraordinarily motivated undergraduates who've worked with us over the years. They would fill up one of these slides easily. Okay, thank you. I look forward to maybe some questions.
Thank you, Brian. That was great. So if you would kindly stop sharing. So we have a question from Mackenzie Lovegrove. Mackenzie is a postdoc currently working with me, who's kindly helping us with timekeeping. So Mackenzie, would you like to ask your question?
Hi, that was a really great talk. I had a question concerning what type of bees you're using in this, whether you're using nurse bees or foragers, and if you see a difference between their capacity for this plasticity you observe based on that, given that nurse bees are often staying within the hive and foragers tend to be out and about and are probably exposed to more novel stimuli?
We use foragers, and we explicitly pick foragers because the learning of bees that are in different behavioral castes can be different. So we explicitly limit that. And I will say, I've got some papers on that. And if you send me an email, I can send you a barrage of things to read. That young bees tend to not learn as well. Guard bees get really angry and they're hard to train. And forager bees are just motivated to learn. And I'll say it's even more specific than that because we're blessed with having wonderful beekeeping facilities at Arizona State. And we can breed bees, and we work with a restricted genetic population. We can't standardize bees like all of you can standardize fruit flies and now mosquitoes, but we do the best we can with it to try to stand, because genetic background has an enormous influence over how they learn. Every learning paradigm but the novelty one talked about today, we have genetically selected bees that learn differently in those paradigms. And we published a paper last year how these different learning phenotypes are really important for functioning of a colony. That was a long answer to your question.
No, it was really interesting. That sounds like a great setup you've got there.
Thanks, Brian. So Zane, you have a question?
Yeah. Hi, Brian. Great talk.
Hey, Zane, how you doing?
Great. So the stimuli, do they have to be external, you think, or the physiology internally can also create this special separation?
You mean the physiology in the brain?
Yeah, in the body, like hungry, angry fly.
Oh, yeah. So I showed modulation from serotonin, dopamine, octopamine, and a number of other things there in the antennal lobe. And some of that feedback, serotonin, could be from an alarm neuron. At least there's some indication of that, for example. So could that now really upregulate a bee's responsiveness to the alarm pheromone? So yes, there are a lot of internal states that can get represented either by the release of biogenic amines in the antennal lobe or potentially even by biogenic amines circulating in the hemolymph that gets into the antennal lobe. So yes, there's a lot there to be revealed. I mean, at first I thought I was going to work on this antennal lobe thing and get everything solved in the space of 5 years, and here I am 40 years later, still working on it. And I'm amazed at how much there is there to work on.
So we also have a question from one of the attendees, Leslie Saul-Gershens asks, what do you think happens when honeybees are restricted to one monocrop?
I think their antennal lobe gets very strongly tuned to that. A postdoc working with me many years ago, she's now at Oxford working on wonderful things with bees. We planted fields of brassica. And so we took over an agricultural plot at State University and just let bees forage on it. And we could show that when you bring the bees in the laboratory, Bertram Gerber did this years ago in Randolph Menzel's laboratory. You bring them in the laboratory, they love brassica and they are focused like a laser beam on brassica. So I think when bees get in this situation where they're foraging in large crops like that, the antennal lobe becomes just extraordinarily tightly tuned to that particular odor. Now, unfortunately, at the time, we didn't have the sophisticated means for doing the physiology and the imaging that we have right now. But if I were to go back to it, I would love to take bees in that situation and look at how their space is, as I've said, warped or curved or deformed, because I think it should, because that's their whole world. But it's just a hypothesis at this point.
Great, thanks, Brian. So it's time to move on to our next speaker. So our next speaker is Yinliang Wang. So he's actually from a university in China, from Northeast Normal University, but at the moment he's based in New York, and so he's speaking to us today from New York. So Yinliang, if you would like to share your slides.
Yeah, thank you. Thank you, Cora. Thank you for this swamp. I'm now in the New York, and she did a swamp for me and to finish it earlier. So today my topic is the ferrohero olfactory sensing of plant volatiles and their potential application in Northeast China. And I have to I will give a brief introduction of the some major biological function of the plant volatiles. The part one is very simplest, repellent or attractant on elm leaf beetles, which is called the Ambrosoma quadripressum. The second part is that the plant volatiles that used as a guiding chemical signal to the overposition behavior of parasitic wasp. The third part is that the plant volatiles used as a collective foraging signal to the larva of the middle mouse. So I will start with very simple way to talk about the attractant and the repellent, about some pests. This pest called the Ambrosoma quadripressum. It's a forest pest in northeast China. It fed only on the elm leaves, and the elm tree is very important in the north part of China. It contributes to the wood industry. It can made of the furniture. Also, the city green belts is used this kind of this kind of trees. And this pest fed on the elm leaves. When the density is very high, the leaves will all eaten up by them and cause an irregular death of the elm trees. So we first screening by the EAG, and we find some chemicals that can elicit the significant response of this elm pest, and we found that one has the highest response, which is called the cyanamide. Cyanamide is from another plant, which is called the Laurus nobilis, and this plant is very interesting. It has some anti-pest function because many of insects don't like the smell of these trees. So we did a binary choice assay. We find in some concentrations it take a repel effect on this elm pests, and we questioned whether it will repel this insects. So we designed some behavioral assay. We make two round-shaped elm leaves, and we made a filter paper, a ring-shaped filter paper covered outside of the elm leaves, and one treated with the paraffin oil, which is a negative control, another treated with cyanamide. And we found that they dislike the leaves treated with the cyanamide. So we consider it as an antiphedant for this elm pest. Then we make a Antennal transcription. We identified several genes, olfactory genes of these insects. And we did some expressing level analysis of these genes. And we found that one of the genes called OBP4 has the most highest expression level in their antenna. So we express and purify the protein. And then we do a fluorescence binding assay on a panel of the post-plant volatiles. And then we found five plant volatiles can binding to this protein. And you can see two of them. One is the dibutyl flate, another is disobutyl flate. All these flate chemicals has a lower Ki value, which means it has a higher binding affinity to this OBP. And then we made a modeling, homological modeling of this OBP. We found that this OBP is higher, similar to the Drosophila OBP lush. We know that the Drosophila OBP lush is binding to the aggregation pheromone, right? They shared more than 30% of the similarity. Then we do a modeling. Then we found that this OBP has two binding sites. One is located between the helix alpha-1, alpha-2, and alpha-3. Another site is closely positioned behind helix alpha-5 and alpha-4 and alpha-5. Then these five chemicals, 4 of them are binding to the site 1, but the cyanomaldehyde also has some small binding affinity, but it's binding to the site 2. We identified as a repellent of the cyanomaldehyde previously. Then we make another calculation. We choose another panel, a large panel of the chemicals to do the dockings, and we found that 2 of them has some higher score, which means it has the potentially binding to these OBPs, which is one is called the butyl octyl flate, another is azo-eugenol. And to make sure of that, we do binding fluorescence binding assay again. We can see that these 2 chemicals all binding to this, truly binding to these OBPs. And we can see that OBP is highly sensitive to the flate chemicals. So we try another electrophysiology and also the behavioral assay. We can see the flate kind of chemicals is an attractant of this pests. So this one is already we developed some field trap that can be used in the field to trap this pest. So besides the repellent and the attractant, there are some more complicated behavioral effect of the plant volatiles. Another case is that is this parasitic wasps, which called the Anastatus japonicus. These wasps can control of the Lychee Stinkbug. Lychee Stinkbug is the major pest of the lychee plant because China has a big amount of the lychee industry. It occupies nearly 1/3 of the world lychee products, and the Lychee Stinkbug is the major pest for the lychee. So we can use this wasp for the biocontrol of the Lychee Stinkbug. We can see the life cycle of this wasp. They lay eggs in the stinkbug's egg, and the larvae become larger and larger, and it fed on the nutrients inside the egg. Then they become the pupa, and the egg dead, and they emerged. So our question is that, is there any chemical signals that can guiding the wasps to find their host eggs? So we first make a behavioral screening of all the chemicals, a panel of chemicals in their environment. So we can see that in the boxes, we put two small plastic boxes there and put some eggs on and put some odorants underneath them and release the wasp to let them to overposition to lay eggs. And we found that some chemicals has an attractive effect, and some can rebel their overposition behavior. Secondly, we also test on this panel of chemicals with EAG to see whether they can sense these chemicals at electrical physiology level. And we choose those both has the behavioral effect, also has the electrical physiology effects. We choose these chemicals as the candidate chemicals for further explorations. So finally, we found 8 chemicals that can both elicit the EAG response, also has the behavioral effects. So we questioned which olfactory gene is participating in sensing of these functional chemicals. So we did another Antennal transcripts, and we identified 48 odorant receptors and 13 anotropic receptors in this species. And first, we want to make sure which pathway they relied on to sensing these biological functional plant volatiles. So some previous studies said that the anotropic receptor also participated in the olfactory sensing. So we first make sure which pathway they are rely on. So we silencing all the co-receptors. First is alkyl, IR8A, IR8A, and IR25A. And we saw that after injected with ES alkyl, their parasitization rate has a significantly reduced, but other groups is not. So it means it's rely on the odorant receptor OR pathway. They also, after the alkyl was silenced, they have to expend more time to find the eggs. And we want to know which OR is turned to these chemicals. So we did RT-PCR to find some that specifically expressed in the female antenna. And we silencing all these ORs to try another EAG. If the EAG is has a significant decrease, well, we can know that this OR is turned to that chemicals, because on that time we don't have the OSI system. So we find OR 35 has a significant decrease of the E-alpha phonesis and beta-carotene. And to confirm this, we did another behavior assay. Again, to confirm this, we can see after silencing of OR35 that the beta-carotene and the E-alpha phonesis, the overposition attraction index has significantly decreased. So that's the conclusion. The WASP relies on the OR-based pathway OR35 to sensing these chemicals and to find their host and lay eggs. So the last story is about meadow mouse. This meadow mouse, the larva of there is fed on the soybean leaves. And one interesting phenomenon is that when the larva density is very high, they eat all the leaves. They need to find new foods. Sometimes it has some pioneers. Only a few larvae can find the new foods. Then a large amount of the larvae follow them. So we question whether there is some chemicals that can guiding them to find the new plant sources. There's another story. We find a pheromone binding protein. I don't talk it too much because I focus on the plant volatiles. So we did on the oocyte, and we find one OR is turned to indole and methyl salicylate, another is turned to 3-octanol. And we did a larva EAG. We can see that the methyl salicylate at all concentration it give the highest response, and also they take a attraction effect on the larva of the meadow mouse.
2 minutes.
And to make sure it, we did a fish on the larva antenna. We can see that it's not well developed. The olfactory neurons are clustered, but most of the neurons are expressed OR1. And we do another silencing on the OR1. We see that it also has a decrease of the EAG response of the MESA. And where this chemical came from, it's not a common plant volatiles. It caused the HIPBS. It means only the leaves is foraged, it can come out. So we did a GC mass on the foraged and unforaged leaves. Also, we did a co-injection to make sure this. So the methyl salicylate is a HIPBS. So to further explore its function, we did a behavioral assay. First one, we used the foraged and unforaged soybean leaves. The larvae are preferred foraged soybean leaves. So we believe there is something happens after the leaves foraged, and the larvae prefer to eat them. And we directly add the methyl salicylate to treat the unforaged leaves. It experiences similar behavioral results like the foraged leaves. So that means we found that compound to guide them to the new food source. And to make sure that we also silencing the DSOR1 and to see their behavioral response, we can see that after silencing, the preference, the foraged leaf preference is disappear. So that's all. So here is my my group in China, and when I did my PhD, I in Walter Leal's lab for 2 years and really learned a lot. And thank you all for paying attention. Thank you for the organization. Thank you all. Thank you.
Thank you, Yinliang. Do we have any questions? And just as a reminder, because I think a lot of attendees have joined since we commenced our section, we can't see if you put your hand up, and so you need to actually write your question into the question and answer box that you will see. That's the only way we're able to see your questions. Are there any questions from the panel members or from the attendees for Yinliang? I might ask one, even though it's a little bit technical, sorry. But how do you did some silencing of odorant receptors. How do you actually do that in your species?
I'm sorry, pardon me.
All right, you did some experiments where you were using double-stranded RNA to silence receptors. Yeah. How do you actually do the silencing, and do you know how effective it is?
I think most are effective because we test the qPCR after the dsRNA injected. In 48 hours, we did the qPCR and we see a significant decrease of the odorant receptor. But some research said that in Leptera, in mouse, the RNA interference is not worked. But for our experiment, I think most of some higher expressed OR has truly some a decrease after RNA interference, but we did on the larva, so it's very effective, I think.
Thank you. Do we have any other questions? I might also ask you, Yinliang, what have you gone to New York to do? Have you gone there to learn something in particular?
I have a position in China, and I go out 1 year for postdoc research. Yeah, and after this, I will go back. Yeah, I have to learn something new and do some new work and learn something new. That's my purpose here.
Can I jump in with a question, Carl?
Yes, we can.
Okay, go ahead.
With your parasitic wasp you have, do they lay multiple eggs in their single host, or is it a one egg, one host type situation? Do you see perhaps a change in the odor profile of that host after it has been injected with an egg at all? Is that something you've looked at, or the attraction?
Yes, thank you. That's a very good question. I think most of cases, this wasp only lay one egg in the egg of the stink bug. It's a one egg layer practice wasp, and this wasp, we also choose some volatiles from the host, from the egg surface. Yeah, but we didn't find it's effective or for this species, we didn't find it effective or can attract or can repel the parasites wasps. Yeah, and we didn't find that. So we only find some plant volatiles can guiding their oviposition. So that's our results.
Yeah, that's really interesting. Thank you.
Okay, thank you so much.
Thank you. So we might move on to our next speaker. And might I also make a request? So some of our speakers who are currently online, if you might share your videos if you feel comfortable doing so, just so that the presenter has some faces to look at. I think that would be really appreciated, particularly some of our younger presenters. The speakers who are online who are from the US, it's now late in your evening and you don't need to share. Any of our new cohort of speakers, I think that the current presenter would really appreciate having some friendly faces to look at. So our next speaker is Helen Rushby, and Helen is at Monash University here in Melbourne. So Helen, if you would like to share your slides. So we've now travelled across the Pacific, finally, in our journey around the world. And our first presenter from Australasia is Helen. So thanks, Helen.
Hi, thanks, Coral. So my PhD project is the role of olfaction in macronutrient balancing using Drosophila Melanogaster. Ooh, slide won't change. There we go. So I'm part of the Mirth lab. So my supervisor is Kristen Mirth. And in general, the lab investigates the mechanisms that underlie genetically and environmentally induced variation in body shape and size. So the majority of an animal's energy comes from ingesting 3 macronutrients, and these are carbohydrates, protein, and fats or lipids. Many animals use their sense of smell to find these nutrients. But when they find these nutrients, then they're faced with really important decisions, like should they eat this food, how much should they eat, and is there something better nearby? And these decisions are really important because the ratio of nutrients that an animal ingests actually affects its lifespan, reproduction, risk of cardiovascular disease, blood pressure, and glucose tolerance. As such, animals will strive to reach an optimal nutrient target, and this behavior is known as macronutrient balancing. One variety of macronutrient balancing is protein leveraging, coined by Steve Simpson and David Robenheimer. And this suggests that nutritionally unbalanced diets lead to the overconsumption of overall food, so that animals will compensate for the deficit in a nutrient by consuming more overall food. So there's actually evidence that humans consume more total energy from ultra-processed foods because they're diluted in protein. And there's evidence that children and adolescents with obesity also protein leverage. So the relative amount of nutrients that are consumed are likely contributing to the current obesity epidemic. Olfaction can also affect feeding behavior and metabolism. So flies will increase their food intake when exposed to odors, and increased odor detection in mice results in greater food intake. Also, the mere scent of food has been shown to change neurons from a state associated with hunger to a state associated with satiety. Further, olfactory deficiencies can result in a lean mouse phenotype, and flies and mice fed high-fat diets have olfactory impairment. So it seems pretty compelling that olfaction is intertwined with feeding behavior and even metabolism. So we know that olfaction can affect how much an animal might eat, and we know that it might affect its metabolism. But I want to understand whether olfaction affects the way that an animal macronutrient balances. So to investigate this, I'm using Drosophila Melanogaster, or the fruit fly. The fruit flies get their nutrients from rotting fruit, which is where they live and breed, and they get their carbs from the sugars and their protein from the yeast from the rotting fruit. There's evidence already from the Mirth lab that Drosophila larvae protein leverage. So what this graph here shows is that 4 different diets are increasing from left to right in the protein to carb ratio. What this shows is that larvae fed the lowest protein to carb diet will consume more food overall, presumably compensating for the protein deficit by consuming more food, therefore protein leveraging. But Drosophila are hollow metabolists, so they have different life stages, so they probably have different nutritional demands depending on what life stage they're currently in. So what about the adult fly? Before I can manipulate the adult fly's olfaction, I need to first characterize how they macronutrient balance normally. So I've been characterizing the way that adult flies macronutrient balance, and I've been using capillary feeding assays or CAFE assays, which involve feeding flies or a fly or group of flies different liquid diets in a capillary tube. I've been using an outbred wild type population called red Dahomey, and I've fed the flies 3 different diets varying in the protein to carb ratios, male and female flies, virgin and mated female flies, and flies of different ages. So what I found first is that the 1-week-old male and female flies balance their food intake in the same way that the larvae do. So this is the same negative relationship between the protein to carbohydrate ratio and how much the flies eat. So in the lowest protein to carb ratio diet, the flies are consuming more overall food to compensate for the protein deficit. And both the males and the females did this in the same manner, same pattern, but the females ate more, which was unsurprising since they need more energy for egg production and laying. And this effect was regardless of the caloric value of the diet. I also have plotted the carbohydrate and the protein intake of the flies. So what this intake array shows is that across the 3 diets, the flies are quite strictly regulating their protein intake to between like 0 and 20 microgram per fly. But you can see that the amount of carbs that they eat is a lot more variable. And so it seems that 1-week-old flies prioritize their getting enough protein at the expense of over or under ingesting carbs. Then I did the same CAFE assays with virgin and mated female flies, and I found that they balance their nutrient intake similarly. As you can see here, same negative relationship between protein to carb ratio and food eaten. And here, the same sort of intake array, we can see that the mated female flies are tending to eat more protein, which again is for their egg production and egg maintenance. Interestingly, I found that the way that the fly's macronutrient balance changes with age. So there was a significant effect of the diet, age, and significant interactions between age and sex and P/C ratio and age, which means that as they aged, the way that they balanced their intake changed. So you can see later on when they're older, they're not changing their intake across diets anymore. And pairwise comparisons actually showed that the females stopped doing this earlier than the males. So by 2 weeks of age, the females are no longer macronutrient balancing, and by 3 weeks of age, then the males are no longer macronutrient balancing or protein leveraging. And then again, when I plotted the carbon protein intake per fly, I found that the females only consumed more protein than the males at 1 week of age. Henceforth, as they grew older, they no longer consume more protein than the males, which I think was interesting because I think the prevailing sort of thought was just that female fruit flies always eat more protein than males, just like at any age, they just eat more protein. But this shows it's only in the first week of their life, and this also correlates with when they lay the most eggs. Well, what might be changing this, what might be causing this change in macronutrient balancing behavior with age? So perhaps it's a decline in chemosensation, because with age comes diminished sense of smell and taste, and also malnutrition is prevalent in the elderly population. So next, I will be altering fruit fly olfaction to understand how changes in olfactory ability alter macronutrient balancing. I've just started doing CAFE assays with anosmic flies. These flies are mutant for ionotropic receptor 8A and 25A, Orco, and gustatory receptor 63A. They were kindly provided by Richard Benton's lab. So Orco is a receptor that's essential for the function of olfactory receptors. IR8A and 25A are broadly expressed receptors that detect odors, and GS63A is a gustatory receptor that's involved with olfactory detection of carbon dioxide. So we can expect that these flies can't smell. So these are very early preliminary results that don't yet really uncover any role for olfaction in macronutrient balancing yet. But behavioral experiments are notoriously noisy, and I'm going to need to do a lot more replicates in order to uncover any effects. My next plans are to introduce odors into the cafe assays while the flies feed. So yeast odors, probably like acetic acid, phenylethanol, ethanol, things that they would be exposed to in the rotting fruit. So to do this, I just embed a pipette tip in the agar while the agar sets and put the liquid odor inside, and this way they can't touch the odor with their legs or smell or their proboscis. So they can't taste the odor, they can just smell it. And next I've started back crossing some flies with the temperature sensitive transgene Shibir. So I'm going to use flies with the Shibir temperature sensitive transgene in conjunction with the Gulf-4UAS system. And this way I can activate and inactivate the receptors of interest by putting them at 29 degrees or lower. Interestingly, I know it's not insects, but I'm also concurrently doing experiments with anosmic mice with Professor Zane Andrews at the Biomed Discovery Institute at Monash Uni as well. So the mice are also anosmic and they are feeding on different P to C ratio diets. So we'll have a mammalian model as well. I suspect I've gone through this too fast. In conclusion, adult flies balance their macronutrient intake in the same manner as larvae. And the macronutrient balancing behavior of the flies changes with their age, and this change in age could be due to diminished chemosensation. The preliminary cafe assay results with anosmic flies don't yet reveal a role for olfaction in macronutrient balancing, but I have imminent plans to also manipulate the olfactory system of the fly and to expose the flies to odors while they feed to further uncover a role of olfaction within macronutrient balancing. I'd like to thank the Mirth Lab, the School of Biological Sciences at Monash Uni, the government for my scholarship, Professor Richard Benton for the anosmic flies, and Coral, Walter, and Wynand for hosting the Insect Olfaction and Taste in 24 Hours Around the Globe Symposium. Thank you.
Thanks, Helen. And no problem at all, because you'll get us back on time if you've been a bit quick, so it's no problem. And so Helen is a PhD student, and so it's really a great opportunity for PhD students to present in a symposium like this as well. So if you'd like to stop sharing the slides, Helen, and we can take questions. If we've got any questions from panel members or attendees, remembering attendees, you need to type your question into the Q&A for us. Just give everyone a minute. We do have a lot of attendees, Helen, I assure you. No questions for Helen? Oh, we've got Wei. Is that a question or a comment? I think it's a question. Would you like to ask it?
Yeah. Thank you. Yeah, very nice talk. So my question is, in your mind, how do the flies sense the nutrient internally and know they eat too much or eat not enough to balance the macronutrient?
Well, I forget what it is. It's known as like GCN2 or something is a receptor that senses the nutrients, at least GCN2, I think. But exactly how they do it, I'm not entirely sure. There is a lot of evidence that animals will just do this in the wild. Like, I mean, obviously we have a lot more influences in our society, but they seem to be able to be more in tune with their nutritional needs.
Okay. Yeah, thanks.
Do we have any other questions for Helen? I'm not seeing any, so I think we might move on, and we're now nicely back on time. But thank you, Helen, for your lovely talk.
Thank you.
Right. So our next speaker is Paul Szyszka, and so Paul is from the University of Otago in New Zealand in the beautiful town of Dunedin, and he's going to talk to us about evolution of olfactory degradation in recently flightless insects. So we'll hand over to you. Thank you, Paul.
Yeah, hello, and thanks for letting me talk here. So, since 2.5 years, I am in New Zealand. Before I was in Giovanni Galicia's lab in Germany. And I did my first field study in my life here in these mountains. So I will talk about a project which I did together with Stephanie Neupert, Graham Collach, John Waters, and Brodie Foster, all from the Department of Zoology here in the south of New Zealand. So there is this hypothesis that flight poses a selective pressure for fast olfaction. So that has been explicitly stated by several authors. I picked these 3 papers because I know some of the authors are in the audience. If you know more or older papers, please let me know. Why does flight pose a selective pressure for fast olfaction? So if you fly, you experience faster stimuli than if you walk. The further you go up in the air, the faster odorants move. The air becomes more turbulent. So the stimuli, the olfactory stimuli, are much faster for a flying animal than for a walking animal. Another reason is, of course, flying animals move faster by themselves than do walking animals. And indeed, insects have a very fast olfactory transduction and olfactory capabilities. For example, this is one example where we recorded the speed of olfactory receptor neurons in fruit fly antennae. So this is an olfactory receptor neuron sitting in a sensillum, and we did single sensillum recordings. We knew exactly when the odorant hit this sensilla. And what we found is that if you increase your odorant concentration, you could make these olfactory receptor neurons respond as quick as within 3 milliseconds. So olfactory receptor neurons can respond extremely fast to arriving odorants. Insect olfactory receptor neurons also can resolve very fast fluctuating odorants. So this is a study I did together with Brian Smith. What we found in Arizona is that it actually didn't matter if we take a honeybee or locust or even these slowly moving hissing roaches. Their antennae all could easily resolve olfactory stimuli which fluctuated more than 100 times per second. So yeah, we learned that olfactory transduction is fast in insects. And we also found that insects use their fast smelling capabilities. For example, this is one example in fruit flies, which we let fly in a wind tunnel, and then we asked them, can you discriminate odorant mixtures where both odorants arrive at exactly the same time, and odorant mixtures where the odorants arrive with a short delay of 33 milliseconds. And so what we did is we measured whether these flies approach certain odors. We found that they approach butanone, which is an attractive odorant for flies, less approached butanal stimuli. And when we mixed those 2 odorants, and by mixing this more aversive with this attractive odorant, we thought we create the situation like, or we simulate like a spoiled food source, for example, where the attractive and the aversive odors come from the same source and arrive at the same time, and then this mixture was not attractive for the fly. But if we introduce a little delay in the onset between the aversive odor butanal and the attractive odorant butanone, and this delay was just 33 milliseconds, then this mixture became attractive again. And so the only difference between this attractive mixture and this less attractive mixture was this introduction of this time delay in the arrival of these odorants. And we found the same for honeybees. Bees could discriminate delays of just 6 milliseconds, for example. Okay, these were a few examples to demonstrate that olfactory transduction and olfactory capabilities of insects have a high temporal resolution. now we have to keep in mind that whenever you want to process information in the nervous system, that this is energetically costly. So fast olfaction is energetically costly because it requires high action potential rates and a low membrane time constant, which are both energetically costly. And so what we predicted is that insects which lose flight lose the speed of olfactory reception. Yeah, similar to these examples of cave fish, which lose over evolutionary times their vision. Okay, and we tested this prediction in stoneflies. So in New Zealand, you have the New Zealand stoneflies, Celandoperla fenestrata, and this species comes in two lineages. The ones which have fully developed wings, they can fly, and then we have populations which have reduced wings and they can't fly. And you find them all over the South Island. And in order to find both the winged ones and the wing-reduced ones, you have to climb up mountains with rivers. And then if you are above the tree line, you find these stoneflies which have no wings. And below the tree line, you find these full-winged stoneflies. The wing-reduced stoneflies have evolved like 15,000 years ago after the last interglacial, and they overlap, and from time to time they even mate. So we find like 5% of those flies which we collect are hybrids from these two lineages. And so we went into these regions to collect full-winged and wing-reduced stoneflies. We did this at 5 different locations, which all had genetically independent populations of those New Zealand stoneflies. And in all these 5 populations, flight loss evolved independently. This is also important to keep in mind. So we measured the strength and the temporal resolution of olfactory receptor neurons by electroantennogram measurements, where one takes a piece of the antenna, puts it on an electrode, puffs odorants at it, and then one can see the summed activity, which reflects the activity of the receptor neurons in this antenna. And we always compared these electroantennographic signals from wing-reduced, always in green, and full-winged stoneflies. Here's an example where we puffed 2-heptanone to these antennae, and what we can see in this population, which we sampled at this Mount Burns, that the wing-reduced stoneflies showed slightly lower antennal responses. We didn't see this for these 2 other odorants. So there we didn't see a difference in the strength of the antennal responses. But now if we look at the other 5 populations, we see, for example, for octanol in the whiskey stream population of stoneflies, that again, the wing-reduced stoneflies showed weaker responses. And so the general finding was that whenever there was a significant difference in the strength of antennal responses, this difference was a weaker response in the wing-reduced stoneflies. Then we looked at how fast do these antennal signals rise, and we found again in some populations and for some odorants, for example here in the Mount Burns population for butanone, that the wing-reduced stonefly antennae, they had a little bit slower onsets. So we find this here, or again for butanone in the 6-mile stream population. And then we looked at the offset.
5 minutes.
And here, yeah, and here we found that also in most cases when there was a difference in the offset time, the wing-reduced stoneflies showed slower offsets. Only in one population for one odorant, the full-winged stonefly antennae showed a slower offset. So, in general, one can say that the antennae of wing-reduced stoneflies show weaker and slower antennal responses to odorants. Which supports the idea that flight poses a selective pressure for the speed of olfaction. Yeah, what I found interesting is that independently wing-reduced stonefly lineages showed degraded olfactory response strength and speed. This suggests that suggests a parallel evolution of sensory degradation. And yeah, this supports the hypothesis that flight poses a selective pressure on the speed and strength of olfactory receptor neurons. Okay, and so I had this. That's me, the sound. I don't want to. So for those who might wonder why does he use stoneflies, which are known to be very bad flyers, and in fact I never saw a flying stonefly. So we use stoneflies obviously because these were the only insects where within the same species we had these winged and wing-reduced lineages, and yeah, yes, they are probably not the best model system. So we also applied fluctuating stimuli to get a better measure of temporal resolution, which we did here. So we presented a 10 Hertz fluctuating odorant stimulus, and here measured it with an odorant measuring device, a physical device, where we can see that our odorant stimulus indeed fluctuates at 10 Hertz. So this is a power spectrum at 10 Hertz. We see increase in the power. The insects that I'm used to work with normally easily resolve these 10 Hertz oscillations. So honeybees, which we recorded here, show that their antennae follow these 10 Hertz, but neither the full-winged nor the wing-reduced stonefly antenna resolved these odorants. So it might be that this poor temporal resolution of stonefly olfactory receptor neuron, it could reflect the poor flying ability of stoneflies. Okay, I hope I didn't distract you too much with this last part. Yeah, thanks to you, and that was my talk. And I'm happy to take questions. So I stop share. Good.
Thanks, Paul. That was fascinating. I have a question, but Mackenzie's also got a question. Mackenzie, would you like to ask your question?
Sure. That was a really interesting talk. Stoneflies are really cool. I did have a question. You mentioned about 5% of the individuals you find are hybrids. Have you had a chance to look at whether they show an intermediate response speed at all, or what you'd predict they might be like?
Yeah, I don't know. So this 5% hybridization, this is data from a genetic study, but I didn't include any genomics data, so I can't correlate genomics to my responses. Yeah, I can't say anything to this.
We've also got a question from Melissa Jordan. Melissa, would you like to ask your question?
Sure. Hi, Paul. Great talk.
Hello.
Hi. I was just wondering, do you think that 15,000 years is enough time, evolutionary time, for these insects to really show that kind of significant difference in speed? I mean, is that why you're seeing just small differences at the moment, and you would expect that over more evolutionary time these will just get bigger and bigger and bigger?
Actually, I don't even know whether it is so short time. So I'm not at all an evolutionary biologist or geneticist. I have no idea. One interesting thing is some of those populations showed clear genomic differences between the winged and the non-winged lineages, and others only transcriptomic differences. But we didn't find any correlation to, yeah, in the difference in the antennal responses. But yeah, so it didn't matter whether they were genomic differences or transcriptomic differences. Yeah.
Thank you.
Wei, you have a question?
Yeah, nice work, Paul. So my question is, did you have a chance to compare the winged and the non-winged, the stoneflies, their antennae and sensilla morphology? Their antennae show the same or different?
Yeah, we didn't do this, but we want to do this. Here in New Zealand, it's really difficult to get access to electron microscopes. It's super expensive, so we are still working a scheme out to do these measurements. Yeah.
Thank you.
So I think Paul Wei's sort of, I guess, starting to get at what the mechanism of this might be. And again, appreciating that you mentioned you're not a geneticist, but given that you can actually make hybrids, there would be the opportunity to actually genetically map what might be the cause. Is that something you're interested in?
Yes, so I'm trying to convince my collaborators to do transcriptome analysis of antennae, and we got a lot of feedback from Melissa, but still it does not work so good in the hands of our geneticists. But we are trying to do this, and also we are a bit restricted, so we only have a short time window when we can sample these flies. But yeah, this is what we want to do in future. I mean, so my collaborators, they found already that there are some odorant binding proteins or odorant removing proteins like Pinocchio, which are differentially expressed in these winged and non-winged lineages.
Thanks, Paul. Do we have any other questions? Or so again, as a reminder, attendees, you need to type questions in the Q&A. We've got one here from Eric. Eric Jane is asking, could the difference in EAG response between populations be due to temperature, whether flightless versus flighted populations live above the tree line versus below the tree line?
That's a good point. I mean, we took them in the lab when they were nymphs, and we raised them in the lab. So we had them in the lab like for a month before they emerged as adults. So in this time period, they all experienced the same temperature. But I mean, yes, in general, I think it's plausible that this could be reflect different adaptations or adaptation to different temperatures. I have to think more about this, but I wouldn't exclude it at this moment.
That's great. It's always great when we get new insights from presenting. So thanks for your question, Eric. Yeah. Any other questions from the panel or from the speakers or from the audience? That looks like we're about done. All right. Thank you very much, Paul. I'm just going to hand over to Wei now, who's going to host the next couple of talks. Over to you, Wei.
Thank you, Carol. My name is Wei. I'm from Murdoch University and based in Perth, Western Australia. And yeah, it's my honor to coordinate this session with Carol and also making it. And first, I want to thank all of you from different countries, different areas, still be with us. Some of you already at night, some maybe in the morning. And so our next speaker is Dr. Nobuaki Takana from Hokkaido University from Japan. Yeah, can you share slide?
Okay. First, I'd like to thank organizers for giving a chance to talk our recent study to such a wonderful meeting. This study is performed together with Hirao Takashi and cedar, and Akejima. Many insects exhibit a series of actions in a fixed order during ritualistic innate behaviors. This is one example that shows the Drosophila courtship behavior. Here, a male orients toward a mate, taps the mate with a foreleg, extends and vibrates a wing unilaterally, licks the genitalia of the mate, and finally attempts copulation. This is just one example that shows the Drosophila courtship behavior. The male carries out each action in a fixed order in response to multimodal sensory cues. Visual and olfactory cues stimulate the male to initiate courtship and progressing to the next step of tapping. Gustatory input received by the foreleg triggers unilateral wing extension and vibration to produce courtship song. Licking provides gustatory information that induces a copulation attempt. Each sensory cue is received by a distinct sensory organ and processed in a distinct primary sensory center. As you know, olfactory input is received by olfactory neurons in the antenna and is processed in the antennal lobe, AL. Whereas contact chemosensory input is received by proboscis and by test neurons in the proboscis and legs. And that information is sent to the ventral nerve cord, VNC, and subesophageal zone, SEZ. Each sensory pathway converges to the P1 neurons, the command neurons that trigger courtship behavior in Drosophila. P1 neurons have been reported to trigger an entire series of courtship actions from tapping to copulation attempts when they were excited by D3-pair1. In addition, increased activity of ASP22 descending neurons was found to elicit multiple late stage of courtship actions, including licking. While ablation of these neurons, the neurons did not change the behavioral order. Activation levels of these neurons are known to correlate with which courtship actions are triggered or their frequencies. However, it still remains unclear how the courtship sequence is determined in the brain. One question was raised: are there any other interneurons that integrate multimodal sensory input besides these neurons? Previously, we reported 29 types of antennal lobe associated neurons labeled with Gelfond enhancer trap strains as shown on the left. Among them, we identified various types of neurons which innervate multiple primary sensory centers. One of them, lateral antennal lobe tract projection neuron 4, LPN4, projects to the border between AMC and SEZ. AMMC is the antennal mechanosensory and motor center and subesophageal zone, as well as antennal lobe. SEZ and AMMC are the gustatory and auditory centers, respectively, suggesting that the LPN4 may integrate multimodal sensory input and send output to the ventrolateral protocerebrum, VLP. The LPN4 showed unique projection patterns in the antennal lobe. Glomerular alveolization were observed in two glomeruli, VA1D and BA1B, and additional branches were in the ventral glomeruli. VA1D and BA1B are innervated by olfactory sensory neurons expressing OR47B and OR88A that respond to fly smells, male smells, and female smells that's revealed by Wynand. Recent study also showed that OR47B olfactory sensory neurons respond to palmitoric acid, which was found in the cuticular hydrocarbons in both males and females of Drosophila. Here, I showed OR47B and OR88A pathways. OR47B and OR88 olfactory sensory neurons target to the antennal lobe, where multiple tracts of projection neurons receive the input. MPNs receive the input and send the output to the maximal calyx and lateral horn, and MLPNs terminate only in the lateral horn. These two sites are the secondary olfactory centers in the Drosophila insect brain. And LPN4, interestingly, don't target to the lateral horn and maximal calyx, but ventrolateral protocerebrum, where PPN1 and BAV3 ascending neurons also terminate. These two neurons send the information of female cuticular hydrocarbons received by legs and project to the P1 neurons there. These anatomical findings suggest to us that LPN4 received fly smell information and integrate olfactory and gustatory or auditory information that are correlated to the courtship behavior. Today, I'd like to present first sexual dimorphisms in the morphology and odor response patterns of LPN4. Second, genetic manipulation of LPN4 altered the courtship behavioral sequence. And third, female cuticular hydrocarbons inhibited the odor responses of LPN4. And later, we'd like to discuss a neural mechanism underlying the fixed action odor during the early stage of courtship ritual in Drosophila. I first show the sexual morphology of male and female LPN4s labeled with NP6107 GAL4, which can induce gene expression in LPN4 specifically. Comparison of its morphology between males and females revealed that while the length of the primary axon were not different between males and females, the sectional area of axon was larger in males than in females, and the number of branches in the protocerebrum was higher in males than in females. The projection areas were also different between males and females. The male LPN4 innervated DA1 glomerulus partially, and superior clump and epaulet region in the protocerebrum, which are not found in female brains. DA1 is the glomerulus where the olfactory sensory neurons that respond to male pheromone, cisvasenin acetate, terminate. We thus analyzed if LPN4 also showed the sexual dimorphism in the CVA response. When we expressed calcium indicator GCAMP6S in the LPN4, male LPN4 showed larger excitatory response to CVA stimulations than female ones, consistent with the finding that only the male LPN4 innervated the DA1 glomerulus. We also analyzed if LPN4 responds to palmitoleic acid that excites OR47B olfactory sensory neurons, presynaptic cell to LPN4, and confirmed that both male and female LPN4 responded to 1% palmitoleic acid in the antennal lobe. But on the other hand, the excitatory response in the VLP, the target region of LPN4, was observed only in males, but not in females. This result may reflect that the LPN4 axon is thicker in males than in females, as the thicker axons are generally more electrically conductive than thinner axons, or that the female LPN4 is inhibited at the presynaptic sites in the VLP.
5 minutes.
As the LPN4 projection area overlaps with that of P1 neurons in the VLP in the protocerebrum, these results suggest that fly odor information of LPN4 is relayed to P1 neurons in males, but not in females. We thus analyzed the function of LPN4 in male courtship behavior. We first blocked the synaptic transmission of LPN4 by expressing tetanus toxin and observed the male behavior in a small chamber. When we paired the TNT expressing male with a decapitated, motionless female in red dim light to exclude visual input during mating, we found that tapping behaviors until the first unilateral wing extension were skipped more slow in the TNT expressing males than controls. The mean frequency of tapping until the first unilateral wing extension was lower in the TNT expressing males than that of controls. Unilateral wing extension was observed in the TNT expressing males without tapping behavior when female odor alone was delivered. Here, when we set a decapitated female in the lower chamber, separated from the male layer by mesh barrier, we found that the ratio of flies that extend their wing unilaterally was significantly higher in the TNT expressing males than in controls, which was not found in males that had their antennae removed or in the absence of females. These results indicate that olfactory input drove this abnormal behavior in the TNT expressing males, as the mesh barrier prohibited the male from tapping the female. And blocking synaptic transmission of LPN4 skipped tapping behavior more during the course ritual. Wing extension without tapping behaviors in TNT expressing animals was not evoked because of hyperactive courtship motivation, as the frequency of wing extension and frequency of cooperation attempts were not significantly different between TNT expressing animals and controls. We did not observe significant differences in moving activities between control and TNT expressing animals, and the males expressing the inactive form of tetanus toxin did not show any aberrant behaviors as shown here, indicating that the phenotype in the TNT expressing animals was not because of a mutation caused by girlfriend insertion and neither by motor defects. These results indicate that blocking the synaptic output of LPN4 caused wing extension without tapping the female. This means that LPN4 suppresses wing extension when the male smells female odors. We confirmed that the LPN4 suppresses the wing extension by activating the LPN4 by expressing D3.1 at 31 degrees Celsius. As was expected, the frequency of wing extension and cooperation attempts were decreased in the D3.1 expressing animals. And this is not because of hyperactive courtship motivation, as D3.1 expressing flies showed the increased frequency of tapping until the first unilateral wing extension. The D3.1 expressing males showed movement activities comparable to controls at 31 degrees, and normal courtship behaviors actually at 25 degrees. The phenotype in the D3.1 expressing animals were not due to motor defects and the mutation caused by girlfriend insertion. In addition, in contrast to controls in which 96% of males showed the first wing extension until the fourth tap, a significantly lower rate of the D3.1 expressing males showed the first wing extension until the fourth tap. We thus concluded that excitation of LPN4 in males suppressed transition from the tapping phase to unilateral wing extension phase. LPN4 was excited by palmitoic acid, a sort of an aphrodisiac odor, but excitation suppressed post-tapping behavior. These results led us to hypothesize that unilateral wing extension is released through inhibition of LPN4 while tapping a female, as gustatory input received during tapping triggers wing extension. Thus, we investigated whether applying female cuticle pheromones to the foreheads inhibits the odor responses of LPN4 by expressing GCAM6S in the LPN4. We found
15 minutes. If you could start wrapping it up, that would be great. Thank you.
Thank you. We found that application of female cuticle pheromone to the forehead significantly reduced calcium responses in LPN4 to the palmitoleic acid in the ventrolateral protocerebrum in comparison with flies subject to application of solvent. So our study suggests that the action odor during the early stage of courtship ritual is determined by the following mechanism. First, while a male orients to a female, volatile pheromones activate the olfactory sensor neurons, which excite multiple tracts of processing neurons in the antennal lobe. Second, MPNs and MLPN pathways cooperatively induce tapping, while LPN4 pathway suppresses expression of wing extension. Then tapping releases inhibition by LPN4 pathway and excites PPN1 and BAV3 ascending neurons. Finally, a wing extension induced after tapping. We finally thank researchers, institutes, and grants as listed here for their support. And thanks for listening. And sorry for the delay.
Yeah, thank you for your talking. So any questions from audience on the panels members? Okay, so just let everyone know you can input your questions from the question and answer, and for panel members, you can use chat. So if we don't have any questions, so we may move to the next speaker, and thank you so much for your talk.
Thank you.
Yeah, thanks. And so our next speaker is Dr. Paul Cunningham. Paul is from Agriculture Victoria Research in Australia. And Paul, hand it over to you.
Thanks, Wei. All right. Okay, so yeah, thank you for giving me the opportunity to talk at this wonderful symposium today. So yeah, my name's Paul Cunningham. I lead a research team for Agriculture Victoria. We are based in Melbourne, AgriBio Centre, and our research is mainly in applied entomology, so integrated pest management, insect diagnostics and biocontrol. So there's going to be a bit of an applied spin to my talk today. And I'd like to start off by acknowledging the co-presenters of this talk, Kevin Farnier and Jesse Hennigan, who did a lot of the research I'm going to be presenting today. So one of the focus areas of our research is developing lures for insect pests, and particularly these lures that are based on host semiochemicals, so host volatile blends for a number of different agricultural pests. And those tree icons show lures that we've developed that are now in our sort of advanced stage of field trials. And my talk today is about a lure that we've been developing over a few years for the Queensland Fruit Fly, which is Bactris triani, which is a tephridid fruit fly, not to be confused with the Drosophilids. These flies prefer ripening fruits on the trees. This is Australia's #1 horticultural pest, huge problem. It's a native insect. It's highly polyphagous, attacking a wide range of fruit and veg, and causes a lot of problems for growers, for the industry, and market access and biosecurity. So a real issue for us. And it's one of the big sort of challenges in developing these lures is really you can only, you're looking for to be commercially viable, sort of 5 to 7 volatiles, and you've got to pick this blend that's going to fool an insect against a very complex background of odors, often including the host plants itself. And this is what we have to face from this perspective. And the sort of the big question is, what's the best way to go about selecting the different volatiles for these lures? And there are, with polyphagous insects in particular, they've got this really wide host range, many different families of host plants. And so lots of different odors and complex odors, many different volatiles in. And even with electrophysiology, where we can narrow it down to, what the insect might be detecting, we still get stuck with this question of which are the best volatiles to select from all of these different potential volatiles. And the fundamental question that's really kind of challenges behind that is, how do these volatile mixtures determine host recognition in a polyphagous insect and host preferences? And there's different sort of theories to how this might be working in a polyphagous insect, whether it's just a sort of a summation of a balance of the volatile attractants and deterrents. If that's the case, then should we be looking, just go out there and look for the strongest attractants and put them together in a blend, remove anything that looks like it might be a deterrent? And should we be looking, for example, just what commonalities are in key volatiles across the different hosts? Or should we be looking a bit more along the lines of host species related volatiles? So if we look at, say, monophagous or more specialist insects, from the behavior, from the looking at olfactory processing, there are these contextual effects that become conveyed by individual volatiles within the host. And the question is, how much is this important in polyphagous insects? Has the olfactory system been shaped by the host species themselves? In which case, do we go out and just look for blends of volatiles within the most attractive host? Or is there some other sort of broader contextual related effects for polyphagous insects that kind of conveys host image to them of a fruit? There's too many hosts for the olfactory system to track as such, so it's really about just recognizing something that's a host and shaping it to some sort of sensible, evolutionary sensible strategy. In which case, is there some sort of mix of volatiles that determine this host image in terms of from different groups of volatiles or with different effects of ripe volatiles, fruity, green leaf volatiles, fermenting odors? Or is it just something else? Is it a mixture of all these and just improving our understanding from that perspective? So back to Queensland Fruit Fly and this lure. So the lure we're trying to develop is for the female flies, and across the tephritid fruit flies, the females are really, there's two odors that we tend to look at of importance to the female flies. One is the odor of these nitrogenous compounds, or the protein odors, and young females require protein to develop to sexual maturity, similar to Drosophila, like in Helen's talk earlier. And they show a strong attraction to these yeasty fermenting odors, and that's very commonly used in these traps. But in Queensland Fruit Fly, and it's been shown in other tephridids as well, this attraction tends to sort of tail off a bit as the females become sexually mature and wanes quite considerably. But the egg-laying females are attracted to their hosts, obviously, to lay eggs. Olfactory cues are important, fruit odor cues, and also visual cues. So if we want to develop a lure for the mated female flies, we probably need to look at fruit odors, and that's where we've been going with our research. So designing this mated female lure, we did some work a few years ago now that was basically looking at behavioral attraction to different hosts, screening volatiles, GCMS work, electrophysiology, making up lots of blends. And long story short, we found that ripening volatiles were very important. And these three ripening volatiles in particular are common across many of the hosts. And we sort of made up various blends of these volatiles, and we found that really on their own they were very attractive, but when you remove them from a blend, that mimics our host plant, host fruit. They decreased attraction. And also you could do things like we injected them into a not particularly attractive host, which is a squash, and it turns the squash into the equivalent attraction to a lovely juicy peach or nectarine. So again, really showing these ripening volatiles are important. So we knew we had to put those in this blend. But what else? Well, we went on a little bit further, a little bit later on, we were looking at yeast fruit fly interactions and these gut-associated yeasts in fruit fly larvae, which emit odors that attract the fruit flies, the adult flies, and can also deter them depending on the yeast species. And also they're important, those yeasts are important from an ecological perspective for larval development. So Kevin went on to do some electrophysiology, looking at 3 of these odors, these yeast odors that actually are common to a lot of yeast and a lot of fungi, and did some work, the EAG work, electrophysiology work on the antennae, and got nice responses from males and females to these 3 yeast odors. But what was interesting was he also looked at detection on the maxillary palps as well, and found that we got some responses, and really strong responses in males, but also in females as well. So that got us to thinking, well, maybe we need to put some of these yeast volatiles into this mated female lure as well. So we developed a number of trial blends that mixed and matched the different kind of categories of these volatiles. So we had one that was just our short esters, those are the ripening odors. We had a blend that has the short and the long esters, so these more fruity odors. And then we had the blend with yeast alcohols, then we had all 3 together. And we loaded these into these polyethylene sachets that that had different thicknesses to sort of regulate the release rates. Put them inside our visual trap, this lad trap, which we'd shown in some other research, was particularly attractive to these flies with these colors, and hung them up in the orchards to try and to see what happened. So we did these trials in a citrus block to start with. Now citrus isn't a super important, super attractive host to the fruit fly, so we gave them a bit of our lures a little bit of a helping hand there. But you can get a lot of flies in the citrus. And as you can see here, if you look at that blue pin there, that's our blend of fruit and of ripening esters and yeast odors. And you can see that was very good. We've got some nice attraction there. That's our regular protein trap. This is the trap here, the visual trap alone with no volatiles, so they've got a nice effect there. And this is the trap with just the ripening volatiles. What was interesting was when we actually added the longer fruity esters, it actually decreased the attraction in both cases. It's interesting to know how those volatiles are working together. Yeah, thanks. So then we went on to some field trials in commercial orchards, and this is again using this blend, this 6 volatile blend of ripening esters and the yeast alcohols. And we wanted to test it against another commercial trap that's available and the proteins traps, but this time we'll give them a bit more of a challenge. We used a pome fruit orchard, mixed apple and pear orchard, and also a stone fruit orchard, which would be a real challenge for them. And that's our trap there with lots of flies caught on it. And this time we dissected the females, dissected out the spermatheca and determined the mating status of the flies to see what we were catching. And in the pome fruit orchard, it was really nice. We've got a really strong attraction, as you can see from our blend. These are the commercially available protein and so the fruit odor trap. So ours was winning by a long way there. But even when we put the lures inside our visual trap, we still got a much stronger attraction. And also caught some virgin females. So that was very good. We're very excited about that. So then we went to the stone fruit orchard, and we still got a strongest attraction to our lure. Those are the commercial lures again, so it's nice. But when we put the other lures inside our trap, in the stone fruit orchard it wasn't as strong, and you can see there's a lot more flies around in stone fruit orchard as well. But our trap was also, and this is a protein inside the trap, and this is our yeast odors inside the trap. We get yeast and ripening odors, and you can see that also they caught a lot of virgin females, and the trap did really well. So this is our trap, this is our synthetic blend, and this is just protein, which is really interesting when you put just a yeast, this is sort of hydrolyzed yeast inside the visual trap. So in the stone fruit, the results were slightly different. But all in all, I say for us, it's very exciting when you actually can get a lure to work in the field. We've shown that this new lure was effective at catching the flies and better than the other lures that were available. But of course, the big question is will it reduce damage for us from that perspective? So we'll be going on to look at that. I think there's some interesting questions there in terms of synergism between potentially between these sort of microbial odors and fruit volatiles in the tephritid flies. Now that's something that I think, because we tend to see them attacking ripening fruits on the trees, there hasn't been as much focus on the microbial odors as say with Drosophila, so I think that's important. And we found that these volatiles are detected by the palps strongly as well as the antennae, and I think that's an interesting question in terms of the odor processing or perception, but also in terms of how we go looking for the right odors to use. And that, you know, although it's very exciting looking at the olfaction side of it, that we've got to remember that the visual cues are very important, and the way that they interact with the olfaction is also something we have to bear in mind when we're developing these different lures and traps. So that's my talk. Thanks. I'd just like to say thanks again to Kevin, who did most of the GCAD work, the lure development, and to Jesse, who is running and coordinating the field trials, and to various other people that we have collaborated with along the way that have helped with this research. Yeah. Thank you.
Thank you, Paul. So yeah, wonderful work. So any questions? Yeah, I didn't see any question, but I have a question. So why your lure can attract, looks like work very well for the mated female than the virgin female? Any reason?
Well, I mean, that's kind of where the virgin females aren't as strongly attracted to the fruit odors. They're much more strongly attracted to, I say, these protein odors, and it's a little bit of, nobody knows exactly how they're obtaining their proteins. I mean, we've had a little bit of look about whether it's related to the fruits themselves, but it seems to be more, the evidence is more out there that they are feeding on the surface of leaves and things as well. So I don't think I think we're getting closer to, yeah, whatever the attraction is for the mated females, but why the yeast, those microbial odors are important. We've done other work which I haven't shown today, which shows really if they get too high, there's too much fermentation, or if there's larval infestation that's high inside those fruits, they'll actually be deterred by them, and that's quite renowned in these flies. So there's some sort of balance that you've got to strike to get it right.
Thank you. Yeah, we received questions from the audience from Professor Richard Newcomb. The question is, nice talk, Paul. Is there specificity in terms of the yeast? Do the flies transmit specific yeast? Do specific yeast have different odors? Yeah.
So yes, yeah. So that the paper that was kind of up there by Alex Piper, who was he's in our group. He did some work looking at these different. He looked at gut-associated yeast. We actually also look further at just fruit-associated yeast, and they certainly do give off different odors, which have different effects on the attraction of the flies. So how that relates to the we looked at larval feeding and survival, and that also differs on the different yeasts. So yeah, I know that work's been done with Drosophila, and I think it also needs to be looked at in the tephridid flies as well. Which volatiles are actually giving rise to those differences, of course, is one of the key questions between the yeasts, because there's a lot of common ones and a lot of different ones. So
yeah, we also received another question. That's from Nisana Pereira. So the question is just wondering, did you do GC-MS-EAD or GC-FID-EAD?
Kevin's done both. So we've got the capability to do both that are linked together on this. But I can't remember offhand which one he did for this particular bit of piece of work. I can always follow that up, but yeah.
Okay, thank you. And any other question? Please ask. So yeah, okay. So yeah, I have another question. So from your queue, yeah, you identified, you found, did you try any other fruit fly to test on other fruit fly rather than Queensland fruit fly?
No, we haven't, but we'd love to. We're very interested in looking at that. I mean, there are many other pest tephridids which have similar host ranges. So I think, yeah, testing them on other fruit flies would be certainly the next step. And we are keen to we have been chatting with some potential people to collaborate on to look at that, yeah, particularly oriental fruit fly and some of the other big fruit fly pests around the world.
Okay. So thank you, Paul. Thank you for your wonderful work. So
thanks very much for the opportunity.
Yeah. So now
there is time to ask your question, Jason, if you would like to.
Oh,
very good. Very good. Thank you. Sorry to jump in right at the end. Paul, nice to see you. I'm Jason Pitts. I'm from Baylor University. I'm really interested in this fruit mimic that you have in the center of the trap, which is not only a shape but a color. And I just wonder, I'm not very familiar with that literature at all. So could you tell me how extensively has that aspect of the trap been looked at, either for the Queensland fruit fly or for other tephridids?
So that design goes way back decades, I think, with Ragolitis, I think the original work was. And there has been a bit of work, but certainly not enough. We did some work. We published a paper earlier on that really just took that trap and looked at different colors and different combinations of different colors. And we came to the conclusion that that particular red sphere in the yellow trap is the most effective. And we also looked at a sort of two-dimensional versus the three-dimensional, and the sphere itself made a lot of difference. So it's much more attractive as a sphere than it was as just a red circle. But yeah, further than that, there hasn't been enough work on it, I don't think. And really kind of, yeah, honing in on what those visual cues are.
Thank you.
Thanks.